Great. Okay. Good. Hi, everyone. I'm Valentina, and I work at the Health Psychology Group at KU Leuven, and I'm very happy to be able to present some of my recent work here. So for those of you who were here last year, this will probably sound very familiar because it is essentially a continuation of that work. But just move on. Okay, great. So most of you don't need any introduction to the vagus nerve, but it is indeed the longest cranial nerve, and its main function is the maintenance of autonomic function. So essentially, it comprises of 80% afferent fibers that bring information from the visceral organs into the brain. However, despite this being its main function, it also has an important influence on neural processing, which is why vagus nerve stimulation is a very successful treatment for pharmacologically resistant epilepsy and depression, for example. However, despite the success of its treatment, it is, of course, an invasive, surgically implanted treatment. So for this reason, ever since its approval, there have been attempts to come up with a non-invasive version. And that brings me to the transcutaneous auricular vagus nerve stimulation. So we are lucky, in essence, that vagus nerve on its way into the brain actually passes by and innervates parts of the outer ear, which means that by placing the electrodes onto the outer ear, we can actually achieve similar effects to vagus nerve stimulation, but non-invasively. And so the main purported mechanism of action for both invasive and non-invasive VNS is upregulation of noradrenergic activity through the locus coeruleus. However, the evidence for this, especially in humans, has been a little bit spotty. So over the last years, there has been a lot of research in both central and peripheral markers of noradrenergic activity due to TVNS, including salivary markers, HRV, pupil size, and neural activity. But it seems that for every positive finding, there is a negative one. So there is a lot of inconsistency and conflicting findings. And it seems that stimulation parameters and other extraneous factors play a large role, which is why there is now also a big push to standardize stimulation parameters and reporting standards across labs. So in this current research, we were interested in resting state alpha dynamics and whether they could perhaps index this noradrenergic increase due to TAVNS. And importantly, even though the vagus nerve is very crucial to this afferent processing of bodily sensations, we still know very little about how TVNS affects interoception. So we were also interested in whether there are any TVNS effects on attentional capture by interoceptive stimuli. And for this purpose, we recruited 45 healthy young adults, and we subjected them to a 2-day stimulation protocol, which essentially means that on one day they had TVNS, whereby we stimulated their Simba concha, which is the portion of the outer ear that is innervated by the vagus nerve. And on the second day, we stimulated the earlobe, which is known to be not innervated by the vagus nerve. And these 2 days were counterbalanced. So on both days, the stimulation protocol was the same, so 45 minutes of active stimulation. And so before and after the stimulation, we measured their resting state EEG to arrive at the measure of alpha power. And during the active stimulation, we delivered paired inspiratory occlusions. So as you can see in this flow and pressure diagram, every once in a while, we occluded their inspiration 2 times within a single inspiration. So that was the interoceptive stimulus. And then as control stimuli, we used paired auditory clicks, essentially to see whether there was any difference between interoceptive and exteroceptive stimuli, because auditory stimuli are not at all vaguely mediated. Good. So for the results, we found no effect on alpha power due to TAVNS. So as you can see, the only effect we find is the effect of time, in the sense that between the first and the second measuring point, alpha slowed down, which is quite expected with these long and relatively boring experiments. Now, as for the respiratory-related evoked potentials, which was the main proxy of attentional capture by interoceptive stimuli, as you can see, there's basically a perfect overlap between TAVNS and sham. So we find no increasing effect of TAVNS on the amplitudes of REP. And you can see also that because we used pair stimuli, we can also measure repetition suppression, and there is no effect on repetition suppression either. And this is quite confusing, and even more so when you see that it actually worked for the exteroceptive control stimulus. So for the auditory stimuli, we see a marked increase in these mid-latency ERPs due to TAVNS relative to sham. So yeah, this is highly confusing, and because I wanted to then look more deeply into this, I also exploratorily looked at oscillatory dynamics, so time-resolved oscillations. And here we do see some effects. So this is specifically for respiratory stimuli, and these are difference plots. So wherever you see yellow and orange, that's actually significant, but again, exploratory, so take it with a grain of salt. But it does seem that there is a difference in induced theta synchronization. So a larger theta synchronization in the TAVNS condition during this more earlier processing time window. And then during the later time window, we also see synchronization in the alpha and beta bands, which could index preparation to get this second stimulus, right? Because they were paired stimuli. So it seems that in terms of interoceptive processing, there are some effects, so that's promising, but there needs to be, of course, more research to confirm this, ideally with some more sensitive methods than what we used here. Just to very quickly discuss what could be the cause of these confusing results in a way. Possibility is that our sample characteristics played a role because, of course, our sample was not very variable and not very representative of patients, for example, with respiratory complaints. Furthermore, there is a possibility that these occlusions, even though they are not perceived as particularly intense by most people, do probably considerably activate the vagus already. So it's possible that we are looking at a ceiling effect in that TAVNS cannot exert much more influence over this inherent afferent activation of the vagus by the occlusions. So in the future study, there may be a possibility to use subliminal loads instead. And finally, as I already mentioned in the introduction, there is currently a lot of active discussion ongoing about what the ideal parameters of TAVNS are, including also what is the best control stimulation site. And so this is still a very young and very active research field, and we still don't have a lot of answers. And with that, I want to thank you, and I want to thank my colleagues who were part of the research team and our funders. And if this piqued your interest in any way, I am very happy to answer any questions during Q&A. And also, if you think of anything later, just contact me at my email. I'm always very happy to talk. And if I may just very briefly plug something, if you're planning to attend SPR in Prague next month, that's Society for Psychophysiological Research, we would be very happy to see you at this symposium that's jointly chaired by myself and Ilse van Diest, where we will go much more in depth about the current state and future directions for TAVNS. So, thank you.
Thank you very much, Valentina, for your talk. I think we have time for a couple of questions. We have the first one from Shusi.
So thank you, Valentina, for your presentation. I was just wondering if you had included some independent measure, like pupil diameter, that might discriminate people that actually respond to the vagus nerve stimulation or not, because there is variability in people. And how did you choose the intensity of the stimulation? If you select a specific intensity for participants or not?
Yes, thank you. Very good question. So we did include an independent measure of salivary alpha amylase, and there we indeed found no effects. So no group effects, but also there was not a lot of variability between participants. So I'm not sure that we could use this this particular measure to identify responders from non-responders, unfortunately. It would be very interesting to include pupil dynamics. As far as I'm aware, there have been very few studies that combine TAVNS with EEG and pupillometry, so it would be a very good future direction. As for the stimulation intensity, we use a standardized calibration procedure. That's something that's encouraged in TAVNS to try to find individual intensity for every participant that at least at the start of the stimulation feels noticeable but not painful. So we calibrate that as 80 on a scale of 0 to 100, where 0 is no sensation and 100 is clearly painful.
Hi, Valentina. Thanks for the talk. It's maybe a stupid question, but just I don't know the method that well. I wonder what type of visceral feeling the stimulation actually triggers. So what do you actually feel next to the electrical activity?
It's a good question, and it differs among people. Most people tolerate it very well and report no visceral feelings at all, but some people do report, for example, dizziness and nausea due to TAVNS.